is Ryan Carnegie, and he's going to continue to the discussion of summer mortality and thinking about sort of a broader view of that too. Down, we'll go forward.
Okay, thanks, Bill, and thanks to Oyster South for having me back. I'm happy to be here to talk more about essentially the same thing that Joey just talked about, but from a different perspective. I'm a shellfish pathologist, so we're coming at this from more of an aquatic animal health perspective and really trying to, figure out where we are with this and what the direction is forward in a more holistic sense. So let's advance this. So in general, we have a pretty good idea of what the sources of disease and mortality are in oysters in our region, and we have two major pathogens. Perkintus marinus is this the parasite we see in the upper part of this graph. This is dermo disease. This is everywhere in the southeast region, occurring from southern New England to Mexico. It can be very prevalent, infecting 100% of your oysters where salinities are above about 10 parts per thousand. It can cause 15 to maybe 30% mortality if I had to ballpark it in adult oysters in the wild, but also in farms that have been around for a couple of years. And this is primarily a disease of the late summer and fall. The other major parasite in the region, Haplosperdium nelsoni, or MSX, is only known from the Atlantic. It's present all the way from the Atlantic Canada to Florida, where salinities are above 10 parts per thousand, but it's generally not as prevalent as dermo because the oysters are generally resistant to this parasite substantially in the region, both in the wild and in aquaculture. Where we see mortality events from MSX, it can be typically 15 to 20%. Normally in the springtime is probably peak MSX mortality, earlier in the spring, April, May, for example. So those were the big ones, and we have a couple other minor pathogens, Haplosperdium costale on the top. This is a congeneric, a cousin of MSX, that occurs only in higher salinity seaside waters, where salinities are above about 25 parts per thousand. It's not very prevalent. It doesn't cause much mortality, and this is only in larger, older oysters. And Bonamia, I know, if you're in the southeast region, you probably hear a bit about Bonamia. This is this other parasite that we've found in recent years in North Carolina. We very, very seldom see this in Chrysostea virginica in eastern oysters, but it's, if you're reading pathology reports, you'll see that we're looking for Bonamia and trying to make sure we appreciate its impact on the oysters. So basically, we know what the sources of mortality are in general that are caused by the major parasites. It's mostly dermo, and that's mostly in the fall. The rest of the year in this region, the oysters, once they've gone, past the nursery stage, they should be doing okay. But they aren't always. So there's triploid mortality. So Joey presented some of the data from Virginia. We've been looking at this sort of more broadly and regionally. The first reports that we were registering in our pathology lab were about 2012. Like Joey said, mostly farms from the lower Bayside eastern shore of Virginia. Mortality in the late spring and early summer on the order of generally around 30%, and these were in market-size oysters, sort of the front edge, the fastest growers of last year's planting were the animals that were displaying mortality. And as Joey mentioned again, 2014 was a very significant year with much more mortality than we'd seen before, about 85%. So this is when this issue really emerged as an urgent concern within the region. 85% mortality in market-sized animals at a time of year when nothing should be killing them. It was a problem. So is it HABs? Is it harmful algal blooms? Well, the mortality is happening where we're studying it most closely in the Chesapeake Bay. It's happening well in advance of the major harmful algal blooms. So there's no relationship between the mortality and the blooms that we can ascertain. Known pathogens, we see nothing in the way of known pathogens in these samples. Is there any clinical disease of any sort? This is a typical sample of oysters that would cross through our lab. I mean, this is how they look all the time. These are magnificent animals in great condition. There's no sign of any acute pathology. The only signs that we do see from time to time, so this is a section of oyster gill, and I'm highlighting some very intense hemocyte infiltration into the gills in this animal. These are the blood cells of the animal that would typically be fighting infection and so on. They're flooding the gills. Down here at the bottom, there's some erosion of the gill epithelium. So this is a problem, but this is not something that we see all the time in the context of this mortality. We see it quite a bit, but we're a bit uncertain as to the link. But what we do know is that this continues to be a problem. We've had more than 100 reports now from growers from Maryland to Louisiana. Typically, these reports are coming out of, it's late spring, early summer, so continuing that sort of temporal aspect to the mortality. Typically, 1-year-old animals, that were planted the year before, the fastest growing sort of front edge of that crop. And it's typical that the animals look great when they come in the lab, and there's no obvious pathogens that are associated with any of this mortality. And again, these are more photographs that are typical of these cases. So, we started looking at this as a major sort of, very abruptly or quickly sort of assembled project at VIMS in 2014, where we acquired a number of animals, diploid and triploid, from some Virginia farmers, and we, planted them to basically track mortality. So 2014 was the big year, so we could sort of track mortality in a more experimental context as we as 2015 rolled around. And this is just some of the data here. I believe the right graph, this is Nassawadox Creek. This was the site that had the highest level of mortality. And what I'm presenting here is interval mortality as we started registering it. On the left, it's basically from March up until May or late May. The second interval is into June, and then the final interval is into August. And the interval mortality here was 20% at most. Our most lightly affected site was Milford Haven here. Much lower levels of overall mortality. But I think what we should note is that the trends are generally similar. There was this temporal rise in mortality in the springtime that passed and dropped down to a lower level of mortality. But I think the interesting thing to consider when we look at this, and this is the sort of a difference in the titles of these two presentations we're presenting, is this triploid mortality, or is this something more? And this is something we've really been trying to get a handle on. Is it really just the triploids that are dying? And for a long time we started out with that. I mean, that was what that was what we were seeing coming into the lab, but that was also largely a virtue of the fact that was all anybody was growing. We didn't have much comparative perspective on what diploids were doing. And it made sense that the triploids might be troubled because this late spring, early summer period, the water temperatures are very warm. The food environment is not terrific once the spring bloom is passed, so their metabolic demands are high. And these triploids are producing, they're reproductively sterile to a significant extent, but that doesn't mean they're not producing any gonad. They're producing a massive amount of gonadal tissue that's just not developing to mature gametes and being released. So that could represent a metabolic burden that they're compelled to maintain at a difficult time of year, and that might just tip them over the edge. So, but from these graphs, the diploid lines are mixed in with the triploid lines. We're seeing a peak in mortality of some level in the diploids as well. Here's just a bit more data from an entirely different study that we've got running, looking at Vibrio and oyster reproduction in the York River, where we have 2 diploid and 2 triploid lines that are related. A diploid with a related triploid lineage and another one, 2 pairs. And again, in this experiment, we picked up a signature of this mortality event in the late spring that dropped into midsummer where there was no mortality, and then it rose again with dermo in the later part of the summer. But again, in here, diploids and triploids are both showing that signature of some level of mortality that's just higher in some places or in some years than in other places. So what is going on with this? So one of the models I think still is that the stress of gametogenesis and reproduction, both in diploids and triploids, I would argue, is probably underlying what's going on here. interacting with high temperatures, reduce food quantity and quality. Once that spring bloom is passed, it's a very, very stressful time. And whether they're triploid and just producing gonad that's not going to develop to mature gametes, or they're producing all these eggs and sperm, that's still a stressful burden that they're forced to maintain. The fact that we do see this gill pathology that I pointed out, it does point to some type of environmental stressor. The question is which one? Across the vast geography of the problem as we're registering it, from Maryland to at least Louisiana, we could envision any number of different environmental stressors that are locally affecting oysters across this broad geographic range. And really, I think where we need to go is a more robust epidemiological approach to try to look across all of these sites, across all of these different types of oysters and strains and lines and ploides that are growing across different modes of culture, who is dying and under what circumstances. Questions moving forward. We've been basically ignoring, we're focusing on protozoan parasites, which is what we typically study, but there could be a microbiological aspect to this that we've been completely ignoring. We certainly know in Europe, and this is a picture of Chrysostria gigas from Europe, certainly in Europe, the Europeans growing Pacific oysters know that Vibrio pathogens of the oysters are causing a lot of the adult mortality they're seeing there. So we need to look more closely in our systems at what might be happening with regard to microbiology. Could there be a nutritional aspect to some of this mortality that we might manage in some way if we thought creatively about it? What we do know is that our systems now are significantly eutrophic in a lot of places and impacted. Oysters that used to be eating benthic diatoms are now compelled to consume dinoflagellates and bacteria. And there's been work done in Europe looking at oysters that are feeding on different nutritional substrates, developing different fatty acid profiles, that they have different outcomes on challenge with pathogens. So this is something that I think we should think about here. And then finally, since I'm just throwing stuff out there, this is something that we're thinking increasingly about is there a genetic basis to the mortality that's something other than sort of variation among lines or families or someone like Joey talked about. And we know in bivalve mollusks, and Bill, this is my last slide, that there's a heavy genetic load in marine bivalves. They have the highest genetic load of any marine animal that's been studied. So what is genetic load? This is this accumulation of these deleterious alleles that arise by mutation and that are scattered throughout the genome of these animals. We know, based on a lot of theory and genetic research, that oysters and bivalves are loaded with this burden. And is it possible that what we're seeing in the context of breeding, for traits that we desire, we're selecting for these animals, we're inbreeding them to some extent? Are we creating a situation where we have these deleterious recessive alleles that are coming together in a homozygous condition and reducing the fitness of these animals, maybe not under good conditions, but when conditions are more challenging, tipping them over into mortality? So I throw that out there really as just food for thought as we move forward, and that's all I have. I'm happy to take any questions if there's time. Yeah. Thank you.
Any questions for Ryan?
Yeah, sorry.
Thank you so much. That was a great and such an important talk. These mortalities can be really devastating. So the fact that they happen in so many different areas, it's along the Atlantic coast, it's along the Gulf Coast, looking at water quality, looking at microbiota, looking at food resources, it's kind of tough to make, to really find even the traces of a smoking gun in a beer-stained haze. Mm-hmm. They're just not there. So I have a hypothesis that I'll put out for you, okay, is that there's a threshold of oyster density required to really make oysters thrive. And if you think about oysters, that has to happen in a reef. Mm-hmm. And a reef is a landscape scale, typically, as maybe not today, but as a landscape scale, feature of these systems. And when you get down to a certain population level that's really low, there's a failure to thrive. And I've seen that on intertidal reefs, I've seen it on subtidal reefs. It's just based on oyster density. So if you want to be a baby little oyster and you want to settle on down somewhere, you're going to settle on down where there's a lot of life going. There's three-dimensional structure, there's a thriving reef, and if it doesn't smell right, there's a lot of data to suggest you're going to try to hold out, right? Because you only have one shot at this. You're going to hold out until you can go to someplace that might be a little bit better. And I think that the failure to thrive could actually be an adaptive response, because a lot of environmental cues, water quality, and for example, can affect everything from dermo and shell parasitism and predation, right? These massive predation, explosive episodes, of all kinds of drills and other animals that frankly come up when conditions are just right for them, but they're also overlapping with conditions that work well for oysters. And it could be adaptive to just say, there's too many of those guys around, and it's not really helpful for us to expand our population and just expand the dinner table. So I'm just wondering if you've seen any data from oysters where there are high densities of natural oyster populations, looking at diploids or triploids from those areas, and whether or not we're seeing the mortalities.
We are not registering anything in the way of mortalities in wild oysters that we're looking at this time of year. So in the wild populations, it's we'll see a peak of MSX mortality where MSX occurs earlier in the spring. We'll see the dermo peak later in the summer. But and we do sentinel deployments looking for at the timing of acquisition of diseases and mortality. Our sentinel deployments are not registering of wild oysters, are not registering anything in this late spring, summer peak. Regardless of density, yep.
Given the worldwide existence of the dermo, why do you think that we have an absence of the MSX in the Gulf of Mexico? Do we have a theory on that?
I think my theory would be that probably the intermediate host for MSX is just absent in the Gulf of Mexico. I mean, surely after 60 or so or more years, the parasite has had a chance to get to the Gulf of Mexico. But there's never been a lot of MSX activity in the southeast. Probably, I mean, Tal and I could talk about this for hours, probably over beer, but yeah, probably because there's just not the right intermediate host for that particular parasite in the southeast, and in the Gulf of Mexico, it might be entirely absent. That would be my hypothesis. It's something not connected to the physical tolerances of the parasite, which would probably do well in the Gulf of Mexico. Just the whole system for transmission is not in place. That other intermediate something, that worm or copepod or whatever is necessary is just not the right one in the Gulf of Mexico. We don't know yet. No. Yep.